Welcome dear students. Hi, how are you? I hope all of you are doing well. Welcome to our YouTube channel Dr. Ausaf Biotech World. Students, today we are going to discuss three important topics and we are starting with the uses of enzymes in biotechnology. As we have already discussed that enzymes, they are biocatalysts. Enzymes, they are biocatalysts and they have got three specific characteristics, three very special type of characteristics. First of all, they are highly specific. Second character is first their characteristic is the specificity. The second character is that the enzymes, they are able to function and catalyze the reactions, they accelerate the reactions under stable normal physiological conditions of the normal room temperature and the atmospheric pressure. And third, the enzyme catalysis is a highly regulated process. And there is a specific category of enzymes which are known as the regulatory enzymes. These characteristics are exploited in the various aspects of biotechnology on a commercial scale and on a commercial level. The students, as we have already discussed, that suppose if I ask you to write the complete equation of a enzyme catalyzed reaction, so you must be remembering that it is the enzyme plus substrate, and in a reversible reaction a transient enzyme substrate complex is formed that is converted to the product. But there are certain molecules, organic molecules, which are similar in structure with the substrate, and because you know that the enzymes are highly specific and every enzyme has got an active site. Suppose this is an enzyme and this is the active site or the substrate binding site where the substrate comes and binds, and then it is converted. First of all, an enzyme substrate complex is formed, then it is converted to the product by the various type of reshufflings, rearrangements of groups and displacement reaction, the redox reactions. The various, in a series of reaction, it is converted to the product. But there is another type, certain other type of the molecules which are similar in structure with the substrate, and we can denote it by I, inhibitor. I is for inhibitor. So there is a competition between the inhibitor and the substrate, and in place of the substrate the inhibitor comes and it binds with the enzyme. And what happens then when inhibitor comes and binds with the enzyme, then it is basically converted to EI. Instead of this ES, the EI is formed, and then this EI is not converted to the product because this I or inhibitor is not the natural substrate of this particular enzyme catalyzed reaction, so it is not converted to the product. So that is this is known as enzyme inhibition. Enzyme inhibition. This property of enzyme inhibition is exploited on a commercial scale, and this enzyme inhibition is basically exploited in a number of ways, and I will give you some examples of this enzyme inhibition. That these inhibitors and these molecules are known as inhibitors. So these enzyme inhibitors, in therapeutics or in the pharma industries, one good example of inhibitors are the antibiotics. Certain antibiotics, they are the natural inhibitors. For example, the first antibiotic, do you know that what was the first antibiotic that was discovered? It was penicillin. Penicillin was discovered by Alexander Fleming. And you know that what does penicillin do? You know, bacteria has a cell wall which is composed of, this is a prokaryotic cell, suppose, and this is basically the cell wall of the bacteria. Okay, I will make a good diagram for you. Suppose this is a bacteria and this is a cell wall of the bacterium. So this is composed of peptidoglycans, which are basically the mucopolysaccharides, the peptidoglycans. So the formation, you know that there is a biochemical pathway which give rise to the production, synthesis or formation of the peptidoglycans. So what does this penicillin do? That it is basically, and there is an enzyme, transpeptidase, which helps in the synthesis of these peptidoglycan cell wall of the bacteria. So suppose this is a bacterial cell, this is the nucleoid, this is the bacterial cell. So what does it do? The transpeptidase, it helps in the synthesis of the peptidoglycan cell wall. What does this penicillin do? It comes and it binds with this enzyme transpeptidase, and it's this penicillin functions as inhibitor so that bacterial cell wall is not formed. So it is a natural inhibitor of this transpeptidase. Because of the blocking of transpeptidase, the bacterial cell wall is not synthesized. Okay. Now let us move to the another very important let us discuss about another very important use of enzymes in biotechnology, and that is the in silico. For example, if we know the active site of the enzyme, then, because you know that enzyme is not like this that we have drawn and we have shown, it is a globular, enzyme is a globular protein. So suppose the active site, it lies within the core of the enzyme. So what does we can do? We can do in silico drug design. In silico means the computational, using a computational drug design. And what does it do in the computational drug design? We can screen out from a large bank of the chemical molecules a particular inhibitor that can come and that we can dock. This process is the molecular docking. This process is called molecular docking. So in the docking, we screen out, we can screen using the computational tools and databases, we can screen out a particular compound that can come and inhibit this particular active site or the substrate binding site, and it can act as a competitive inhibitor. So in silico, this is known as the in silico drug design. So we can produce the artificial synthetic antibiotics or synthetic drug compounds. So this is the second application that we can make drugs, computational drugs, and this will basically help in the formation of synthesis of various chemical compounds that can be used as drug molecules. Fine, students. The third point, the enzymes, the third application of enzymes in biotechnology is that the enzymes are used as the molecular scissors in the research laboratories of biotechnology. The genetic engineering and recombinant DNA technology, and you know that there are certain enzymes which are restriction enzymes or restriction endonucleases, which are used for the purpose of the genetic engineering in the biotechnology research. Genetic engineering and RDT is recombinant DNA technology. So these molecular scissors, these are the endonucleases, means they can recognize a particular restriction site, having the palindromic sequence and the particular restriction site, and then they can cut a particular site within the DNA molecule, leaving the two type of end, the blunt end, the sticky ends. So because we do not want to go into detail, the fourth application is in the food industry. It is food industry, and the food industry, we use several enzymes. For example, one of the enzyme that is used in the food industry, it is papain. You must be knowing about the raw papaya. That the fourth one is the food industry. There is an enzyme which is known as papain. Papain is derived from raw papaya. It is proteolytic enzyme. Okay, fine. The papain is proteolytic enzyme, means it cleaves and it makes it just like the molecular scissors, the endonucleases which cleave the DNA, they cleave our proteins and they cut the proteins into pieces, peptides and amino acids, and this is on a commercial scale. This papain, it is used for the tenderization of meat tenderization. Tenderization means to make meat tender and edible, to make it soft, because you know that meat is basically considered as a very hard substance. You must be knowing that in the various delicacies, especially the Mughlai cuisine, there are certain delicacies, especially of Awadh. For example, in Lucknow, the tunde kebab are famous, or in that belt, the gilavati kebab, they are famous. In the recipe of these gilavati or tunde kebab, the raw papaya is used for the tenderization of meat. So on a large scale, on a commercial scale, we use the papain, which is an enzyme. Likewise, there are certain other enzymes which are used in the pharmaceutical industries as a diagnostic tool. You must be knowing that there is an analytical tool whose name is ELISA. So ELISA is a technique, full form is enzyme linked immunosorbent assay. This is the full form of ELISA, enzyme linked immunosorbent assay. We cannot go into the details of ELISA, but it is an analytical technique in which a particular type of the protein, it is detected with the help of the antibodies, and the antibodies are linked to the enzymes, and the enzymes are basically attached to the solid support, and when the particular antibodies which recognize the particular antigens or the proteins or antigenic determinants, there is a color change. So in the diagnosis of various viral diseases like HIV, the ELISA test is used, in which particular enzymes are used in the assays. The second is the therapeutics. in the therapeutics also, means for the treatment purpose, for the purpose of cure, many enzymes are used. For example, the strepto, in the cerebrovascular diseases, there is an enzyme which is known as streptokinase. This enzyme is basically used in the cerebrovascular disease, means after a stroke, when there is some accident and there is the in your cerebrovascular system, the blood supply of the brain or some part of the brain is stopped because of the hemorrhage or some other, it is known as a stroke or cerebrovascular disease. So this streptokinase is given, which basically helps in the removal of the blockage of the blood. So this was one topic. Now we are going to start another very important topic, and that is now we are going to discuss another very important topic, and that is the lipids and biomembranes. Lipids and biomembranes. The barriers. Lipids and the biomembranes, the barriers. This is another very important topic that is basically essential for our various type of the courses. And as we know that lipids, they are a diverse group. What are lipids? In the first chapter of biomolecules of this biotechnology book of CBSE class 11th, we have already discussed about the fatty acids which constitute the lipids. Now what are lipids? The lipids, you know that they are a diverse group of non-polar compounds, non-polar organic compounds which are not soluble in water. You know that they are not soluble in water, but they are soluble in the organic solvents, soluble in the organic solvents like the chloroform or benzene. So they are a very diverse group, but some lipids, they have got very special characteristics, and one special characteristic of the lipid is that because they are biologically very important, and they have got there are a number of classes of lipid. For example, the fatty acids, the triacylglycerols, the simple fats and the compound fats. The fats, they are also class of lipid. Then the unsaturated fats and the saturated fats, and the steroids, the steroid hormone, the cholesterol, these all are included in the classification of lipids. But here we are more concerned about the lipids as component of the biomembrane, biological membranes, the cell membranes. Okay, so students, some lipids, especially the lipids which are component of the biomembranes, they are highly specific. They are having some very important characters, and the character is that though they are non-polar in nature, but some type of the lipids, for example the phospholipids, which are the phospholipids, which are components of the biomembrane, they have got a very special character. And the character is that in the same molecule there is a polar group and there is a non-polar group. In the polar group, the polar part of the same molecule, it is the water loving part or the hydrophilic part. So suppose this is the phosphate group, this is polar, it is hydrophilic, and These are two fatty acyl chains, long hydrocarbon chains. They are known as, which are terminating at the methyl group, they are known as the fatty acyl chains. So these two fatty acyl chains, they are non-polar or hydrophobic. So in the same molecule, this is a non-polar part, sorry, this is the polar part, and these fatty acyl chains are non-polar. The polar part is hydrophilic and the non-polar part is hydrophobic. All students, all such molecules which are both hydrophobic and hydrophilic groups in the same molecule, they are called amphipathic molecules. Amphipathic molecules, and or there is one synonym also, amphiphilic molecules. Amphiphilic or amphipathic molecules are those molecules which have got both the polar part and the non-polar part, the hydrophilic part and the hydrophobic part in the same molecule. Fine, got it? So this character of phospholipids, it makes it's very suitable. The phospholipids are very suitable biomolecule which can become and which can constitute the biological membranes. Biological membranes means all the cell membranes, including the plasma membrane, the membranes of the organelles, and the membranes of the prokaryotic cells. These are all composed of a lipid bilayer, and a rough structure, a rough design is like this. This is basically the fatty acyl chain which are linked to the polar part, and there are certain proteins also which are known as, these two type of proteins are there. These are the transmembrane proteins which are called the intrinsic proteins, and these proteins are known as the extrinsic proteins. This is the non-polar core or the hydrophobic core, and this is the head part, the phosphate part. It is the polar head, the polar part, the polar one. This is also polar, the hydrophilic, and these proteins are the extrinsic and the intrinsic proteins. This is the hydrophobic core, and in the mammalian and especially in case of animals, this bilayer does exist, but in case of animals, especially in mammals, there are cholesterol molecules which are present between the non-polar part. Why they are present? In order to make the bilipid layer more and more polar. In order to increase the fluidity of the membrane. So they are present in order to make the more elastic, more fluid in nature, because it is basically a fluid, the membrane, the nature of the membrane is fluid. Okay, bachcho. So that is the thing that these are the transmembrane protein, the intrinsic proteins. Okay, fine, intrinsic proteins. I hope it is clear. And then it gives the strength, the protection to the cell, to the organelle, because it is a bilayer, and then it has got both the polar and the non-polar part. Now we will discuss about the functions of the biomembranes. Functions of biomembranes or the cell membranes. There are many functions of biomembranes, but we will discuss only the fundamental three functions. That it is the biomembranes, they are having the dual nature. Dual nature means both they are composed of the amphipathic molecules. I have explained what are amphipathic molecules. That is why they are a very good, they function as the ideal barriers of for intra and extra cellular transport. Means they function as a selective barriers. They are the membranes, they are selectively permeable. Selectively permeable. What does it mean, the selectively permeable? It means that only the specific molecules, ions, groups, they will be allowed to cross this non-polar barrier. Okay, and there are various types of the transport. If you are interested in learning the various transport processes like the active transport, the passive transport, osmosis and diffusion, the facilitated transport, the shuttle and the various type of the movement of the charged ions, then you can go and watch my another lecture on the YouTube, transport across the cell membrane. It is on another channel, Study Khazana. So if you write my name, Dr. Syed Ausaf Ali, and Study Khazana or transport across the cell membrane, there is a 1 hour lecture. So I will not go into the much detail of these processes. And another very important function is the movement of water molecules, water soluble molecules like amino acids, sugars, so for the first function is that suppose this is a cell, any prokaryotic or eukaryotic, the inside material cannot go outside, the outside material cannot go inside. The second is that because the amino acids, sugars, ions, salts, they cannot permeate the membrane, they cannot enter, go in or out, so there are specific transporter molecules which are proteins. The transmembrane proteins, they are known as the transporters. So for sugars, amino acid, ions, there are specific transporters. For example, there is sodium, potassium, ATPase. This is a transporter which pumps in and out the sodium and the potassium ions, which is responsible for the membrane potential in the neurons. So it is a transmembrane protein or transporter protein. So another function of the plasma membrane is that it has got the transporter molecules which facilitate the entry or exit of the various water soluble ions and molecules. The third one. The third important function of the cell membrane is that it carries out, the third function is the various processes, they are carried out in the plasma membrane. It is not a static structure, it is a dynamic structure. So the cell-cell addition, it helps in cell-cell addition, it helps in the cell signaling or the signal transduction, and the energy related processes, the energy generation, or the, for example, the oxidative phosphorylation. So all these processes, they are carried out, means how the cells, they adhere to one another. It also helps. Then the signals comes, the secondary messengers and all, and the various type of the ligands and the antigens, and they transduce their signals inside the cell. It is also one of the function of the plasma membrane. Then the energy related processes like the oxidative phosphorylation, which is carried out in the inner membranes of the mitochondria, or at some other place, the intermembrane space in the prokaryotic cells, in which the oxygen donors, they donate electrons and they are accepted by the electron acceptors like oxygen, and the energy released is used for the phosphorylation, means for the formation of the ATP. Okay, the ADP plus pi ATP. For this reaction, the ATP molecule is synthesized. So this is a very good and very important, some of the important functions which are carried out by the cell membranes. So students,